Hello, I'm Sandy Deans, Director of GenQA, and I'm delighted to welcome you to the Genomics Quality Assessment Focus on Molecular Genetics webinar. We shall start with a presentation from Farah Khawaja, who's the Deputy Director for Molecular Genetics at GenQA. And then we'll follow with a live question and answer session with Farah, Becky Tracy, who is also a Deputy Director for Molecular Genetics EQAs, and Carl Fratter, who is the Chair of the GenQA Specialist Advisory Group for the Molecular Genetics EQAs. Now, before we start, and as we have many people attending, please can I ask you to mute yourself? And if you have any questions, please add them to the questions box that you will see on the right-hand side of your screen, and we shall address them at the end in the live Q&A session. So I now shall pass you over to Farah to give her presentation.
We shall begin by discussing how the EQAs have been adapted to meet participant needs. I shall explain the changes to EQA formats, and then we will look at the GenQA response following the impact of the COVID-19 pandemic. We'll then review the Molecular Genetics 2019 EQA poor performance and discuss fundamental EQA issues surrounding these. In 2017, 28 EQAs were offered. 25 of these had samples distributed, and 3 of them were interpretation only. In 2018, there were 20 EQAs, and we introduced panels, so 19 had samples distributed and 1 was interpretation only. Panels included hypertonic infant and polyposis. In 2019, we had 25 EQAs, 24 of which had samples distributed and 1 interpretation only. So in addition to expanding the number of panel EQAs, which are marked with an asterisk in this slide, we also expanded the scope of EQAs, so the number of target genes per EQA were increased. For 2020, 28 EQAs were planned. 26 of these would include samples being distributed, and 2 were interpretation only. These also included 5 new EQAs: epilepsy disorders, osteogenesis imperfecta, inborn errors of metabolism, respiratory disorders, and renal disorders. The change in the interpretation-only EQA involved the pathogenicity of germline sequence variants, for which there would be 2 versions available. One would be classification only, and the other requiring both classification and interpretation of the variants. The number of panel testing EQAs was also increased. In 2019, arrhythmia and cardiomyopathies were 2 separate EQAs. These were merged to form the cardiac disorders EQA. The GenQA website provides information on the disorders and gene targets covered by each EQA. Similarly, Lynch syndrome and familial adenomatosis polyposis, which were separate EQAs, were merged and this year offered as familial colorectal cancer and polyposis. The Rett syndrome EQA was expanded and an epilepsy disorders EQA offered. Multiple endocrine neoplasia and von Hippel-Lindau disease have been offered as separate EQAs in previous years. This year, this was offered as a familial endocrine tumor predisposition disorder. These are just some of the examples where panel testing has increased and the scope of testing expanded. To accommodate the increase in disorders and gene targets in panel EQAs, the number of cases for these EQAs has been increased from 3 to 4. Some non-panel EQAs with multiple diseases have also had their number of cases increased to 4. Another change to 2020 EQAs includes publishing genotyping results following the assessment, so this is prior to release of the pre-appeals report. This will enable laboratories to identify any critical errors and ensure that no patient has been impacted. In March 2020, the COVID-19 impact meant there was increased pressure on laboratories. reduced staff due to illness, having to self-isolate, being redeployed to other laboratories, or having childcare issues. In some laboratories, equipment was moved to COVID-19 testing laboratories. Laboratories were made to deliver COVID-19 testing, and there were supply chain issues. Laboratories have had to reorganise the way they work, adhering to government guidelines for social distancing and personal protective equipment. So how has GenQA responded to COVID-19? As described earlier, 28 EQAs were planned for 2020, 26 of which were sample EQAs and 2 interpretation only. These were planned to be distributed in 2 batches, 16 in early May and 12 at the beginning of June. Taking into consideration the burden on testing laboratories, GenQA have reduced the number of EQAs to 21, so 11 sample EQAs and 10 interpretation-only EQAs. So there's less testing required by the laboratories. These will be split over 4 distributions, starting from 6th of July, where 5 interpretation-only EQAs will be sent initially, then 5 sample EQAs at the end of July, followed by 5 interpretation-only EQAs mid-August, and finally 6 sample EQAs will be distributed in early September. There is a decrease of 7 EQAs from 28 planned initially for 2020 to 21 EQAs. 8 EQAs have been merged, accounting for the decrease in 4 EQAs. So cardiac disorders has been merged with familial hypercholesterolemia, familial colorectal cancer and polyposis with hereditary breast and ovarian cancer, Huntington's disease with neurodegenerative disorders, muscular dystrophies with Charcot-Marie-Tooth disease and related sensory and motor neuropathies. And the remaining 3 EQAs have been postponed to 2021. These include familial endocrine tumor predisposition disorders, osteogenesis imperfecta, and respiratory disorders. Instead of 4 cases, there will now be 2 cases for all EQAs, except MCC and variant validation, for which there will be 1 case. This is because each case for these 2 EQAs has more than 1 sample required to be tested anyway. I'd like to take this opportunity to thank the GenQA sample sourcing and validation laboratories who have continued to help with EQA preparations despite all the difficulties they're facing. Now we will take a look at the 2019 Poor Performance Molecular Genetics EQA cases. In 2018, there were 11 genotyping errors. In 2019, this increased to 31 genotyping errors. There were 2 clear spikes in 2 EQAs, one being the breast cancer EQA and the second in polyposis EQA. The poor performance in these 2 EQAs accounted for 71% of the errors. In 2019, the hereditary breast ovarian cancer EQA had 1 case in which the BRCA2 gene had a deletion and insertion. 13 laboratories reported the deletion only, and 2 laboratories reported no pathogenic variant detected. The 2019 polyposis EQA had a case in which 1 sample had an APC mosaic and MUT-YH pathogenic variant. 6 laboratories reported the MUT-YH pathogenic variant only, and 1 laboratory reported no pathogenic variants in the APC gene. In their report, they did not state the limit of detection and the fact that mosaics could not be detected. 2 marks are assigned for the genotyping category. One fundamental question is if there are 2 pathogenic variants and 1 is missed or incorrectly reported, should 1 mark be deducted and 1 mark retained for reporting 1 allele correctly. For genotyping, if X-linked homozygous, there is 1, or if autosomal, there are two alleles. The consequence of missing one pathogenic allele in a recessive case can be as significant as missing one pathogenic X-linked allele in a male, i.e. a misdiagnosis. In an autosomal dominant case, we don't retain one mark because one of the two reported normal alleles is correct. It's worth noting that if the missed variant is not within the scope of the laboratory's test, and this is clear from the report, then the result is correct within the limitations of the test and the laboratory would receive full marks. The next fundamental question is whether or not miscalling constitutes a genotyping error, even if the consequence of the miscalling is the same. In the hereditary breast and ovarian cancer EQA, 13 laboratories reported a del-A instead of a del-INS in the BRCA2 gene. Another laboratory for the ataxia EQA reported del-T instead of del-INC in the SPG7 gene. So what are the consequences of miscalling? Specificity. This raises concern for specificity of the analysis pipeline used and the ability for complex variants to be correctly identified. With some indels, the consequence of the deletion-only miscall could be very different to the correct consequence. Also, if the report is sent to another laboratory, a specific test for the wrong pathogenic variant could be set up, especially if no positive control is available. Our next fundamental question: does incorrect sizing of a normal size allele at a polynucleotide repeat locus constitute a genotyping error? In the Huntington's disease EQA, a normal size allele of 17 repeats was reported as 21 by one laboratory. So what's the consequence of incorrect sizing? The Huntington's disease marking criteria allocated 2 marks for the statement 1 HTT allele in the normal size range and 1 intermediate sized allele. Information included in a report should be correct. If no size was stated, there would be no penalty. However, the laboratory would receive a comment that the majority of laboratories do include sizing information. In previous EQAs, borderline pathogenic ranges have been distributed to highlight the importance of size. These laboratories need to review their sizing methodology. Incorrect reporting of results cannot count as typographical errors, and we have received appeals in the past stating that it was a typographical error. The incorrectly reported allele was outside of the plus and minus 1 uncertainty of measurement for a normal allele. If an incorrect result is used to test another family member, it could have other consequences, for example, questioning familial relationships or unnecessary resampling. Finally, we ask, should laboratories be able to detect low-level mosaic alleles? For the polyposis EQA, there was an APC variant present at 11 to 13%. The consequence of not reporting mosaicism. Where the variant was not detected, reports did not state the limitations of the test, i.e. not being able to detect mosaicism. Therefore, they gave an incorrect conclusion of a diagnostic report that no APC pathogenic variant was present. Mosaicism at the APC locus is widely documented as a cause of FAP, so it's important to acknowledge these may be missed if the test is unable to detect low-level mosaics. The referring clinician should not rule out APC as potentially causative gene in isolated cases. It's worth noting that it's not essential for every diagnostic APC test to detect variants at low levels of mosaicism. NGS may require higher read depths that is economically practical for routine sequencing. So what's the big deal? The approach GenQA takes to assess genotypes is to ensure accuracy to HGVS guidelines. Consequences of incorrectly reported variants could be severe, impacting on the care of the patient and other family members, so corrective action is required. There's also a wider impact through erroneous entries into globally used database resources for interpreting variants. In conclusion, if a variant has been typed incorrectly on one occasion, there's likely to be an underlying problem in the methodology. The consequence might be negligible in the EQA case, but the next time that methodology gets it wrong, there could be severe consequences for a real patient or relative. The aim is to stress the severity of the error and ensure that corrective actions are implemented. And laboratories are more likely to act if 2 genotyping marks are deducted rather than 1. And GenQA follows up with root cause analysis review.
I hope that talk was useful. Now we have the question and answer session, so feel free to submit any questions as Sandy described using the dashboard on the right hand side of your screen. I'll give people a minute or so to start typing. There's a few already coming in, which is great. Carl, I've got one here for you. Can you hear me?
Yes, I can hear you, Farah.
Right, there's one here for you. Do you have any recommendations for where to check correct nomenclature?
Okay, thanks for that question. So I guess first of all, it's just worth highlighting that it's important that nomenclature is correct and standardized, because that has implications for patient safety. For example, if it's not clear where the variant is, what the exact variant is, then other laboratories might carry out family testing for the wrong variant, etc. So that's one of the main drivers behind standardizing variant nomenclature, and also submission to national and international variant databases. Again, everyone needs to be sure that the variants are correct and unequivocally assigned. So in terms of where to check correct nomenclature, the best place really is the HGVS sequence variant nomenclature website, so the Human Genome Variation Society's website. That's the official website providing the international standard for sequence variant nomenclature. It's not the most user-friendly website, and I would just probably point out that you can also submit questions to the sequence variant description working group of HGVS via the website. I think there's a contact us and an email address, and we as GenQA scheme organizers and EQA scheme organizers have done this on various occasions in the past to clarify nomenclature. I guess beyond that, just briefly, if people are wondering where you would want like tools, web tools and things and software to check that your nomenclature description is correct, there are obviously various ones out there, but GenQA haven't evaluated those, so we can't really endorse anything. But I guess Mutilizer, for example, is a well-known web tool, and Alamet tries to use HGVS nomenclature. But I would just comment that you should always try and check that variants are called in accordance with the guidelines, the HGVS guidelines, and not rely solely on a web tool, probably. Okay.
That's great, thank you, Carl. There's another one here. It's how do you choose the samples that we use for EQAs? So I'm happy to answer that. So we look at issues from previous years that we wish to address to ensure any performance issues and subsequent guidance that we gave to participants, it's been acted upon. One limitation we have is that we use real patient samples. So it really does depend on the sample availability, and it's important that the EQA material we use is as close as possible to real testing material. We try to use interesting cases that will be educational, and if we know about any issues with particular methods or tests that are out there, then we create an EQA scenario or use samples that we know will address these. Got another one here. How do you decide which new EQAs to set up? Becky, can I give that to you?
Yes, of course. Thank you, Farah. So there are a number of things that we take into consideration when we are thinking about how or which new EQAs we're going to set up. The first one is that we try to respond to participant feedback. So this may be emails that we have received where people have said they're particularly interested in an EQA for a certain disorder, or discussions that we've had with other people at conferences who've come up and said, you know, we're really interested in this disorder, do you offer it? And also recommendations from the individual SAG groups and members. And as far as the molecular genetics EQAs go as well, we looked at what was recommended on the NHS genetic test directory as well, to see if there were things on there that we should be offering as an EQA too. So it's very much a larger decision, looking at everything overall and trying to meet the needs of all different laboratories, whether they be large or small as well.
Fantastic, that's great. Thanks, Becky. We've got another question here. So as we said, Carl was the chair of the SAG, and there's a question, what's the function of the SAG? So Carl, you're probably the best to answer that question.
Okay, thanks, Farah. I'll try and answer that. So what is the function of the Specialist Advisory Group? So essentially it provides oversight, advice, helps deliver the EQA schemes that are delivered by GenQA. So in this particular SAG is the molecular genetics core diseases one. So for the group of diseases organized that organizes those schemes. So the SAG's comprised of GenQA senior staff and then about 20 molecular geneticists approximately from different laboratories, and also a consultant clinical geneticist as well to represent clinical input, to provide clinical input. So the members of the SAG work with GenQA staff to help source the samples that are needed for the schemes, to validate the results, hopefully, if their laboratory can do that, put together the clinical cases, the clinical questions that comprise the EQA, prepare and review the marking criteria for a specific scheme, undertake the marking, which is always done by at least 2 assessors, and we get external second markers and things in for that as well, and then writing the scheme reports, reviewing the appeals as well. So as a SAG member, you would generally undertake those things that I just went through for 1 or 2 schemes with the support of the GenQA staff, of course. It's important to note that reviewing the appeals is done by the whole SAG, so that helps provide a wide review with external input, not just GenQA staff. So the whole SAG's involved in the appeals process, and just taking it a bit more widely as well, rather than the organisation of the schemes each year, the SAG also advises GenQA on sort of overall design and operation of the schemes, so future planning, how to develop the schemes provided, how to feed back to participants as well. So I think that really hopefully covers the main functions of the SAG.
That's great, Carl. Thank you very much indeed. So I would just like to continue to say, please do submit your questions. We've got quite a few coming through, so please do. We're here to answer whatever you want to ask us about the EQAs. So here's one, Farah, I'll come to you. So about the marking criteria. So why does the marking criteria change? I guess that means year to year, or maybe across the disease EQAs.
Okay, thanks, Andy. Well, I guess it depends if they're, it depends on the professional guidelines available, especially if new guidance has become available as well. Also, if we set up a new EQA, then we tend to be a little bit more lenient and we make comments rather than deductions, because the idea is we would like to promote the educational element of GenQA. Also, sometimes we do deduct because participants, they respond better to deductions than comments. So when we especially notice that repeated errors are being made, we do tend to make deductions. And the thing is, we've been working hard over the years because we want to just standardize the marking, particularly across similar EQAs, for example, like STRs, and our marking criteria is peer ratified. We use the SAG members to provide to review everything. So we do make sure across all the EQAs they are the same.
Thank you, Farah. It's really important that we work towards consistency as well, isn't it? That's something that the SAG is very keen on. Okay, Carl, can we come back to you? There's another question about not quite the SAG function, but what does being an assessor involve?
Okay, thanks for that question. So I guess assessors fall into two categories. You've got the members of the SAG, and then you've got second markers. So both of those categories of people are assessors really involved in assessing the EQA returns. So I've kind of gone over what the members of the SAG do in terms of organising the scheme and setting the questions, etc. Should probably just add that they will obviously, members of the SAG would only organise schemes that they have expertise in. So they're expert advisors really for a particular subset of molecular genetic disorders where they have the expertise. So as I mentioned before, all returns get marked by two people, so two assessors, two markers. And so the SAG member or a second marker who may not be a member of the SAG, but maybe someone who's volunteered to act as a second marker or assessor for a scheme will mark all the returns against the marking criteria that have been predefined as part of the process. So that's really the main role of an assessor. Is most of our schemes are annual, so once a year in the autumn typically. Obviously, Farah's gone through the changes to the timetable this year, but they would be marking the returns and liaising. The two markers would then liaise at a meeting or virtual meetings, particularly this year, to agree their marking. So mark independently and then compare and talk through the differences, and then bring queries to the wider SAG where it's not clear. And then they would also be involved in writing the scheme report. So that would be done by the SAG member, but the second marker would typically be involved in commenting on that and contributing to that scheme report as well. So I think that covers what the assessors do, hopefully, but feel free to add something else, Sandy or Farah, if you'd like.
No, I think that covers everything. Thank you, Carl. Can I ask just a follow-up question? Just and be nice. What do you get out of being an assessor? Because you've been working with GenQA and also UK EQAs for molecular genetics for many years now.
Have I?
You have. What makes you continue to be involved? What do you get out of being an assessor?
Well, one of the big things, particularly in the first few years of doing it, was I think seeing the returns that come back from different laboratories in the UK and around the world gives you a real insight into how much variety there is in how results are reported, and you can always learn something from where labs are reporting things well and have good wording or good ways of laying out their reports, or and those kind of things. So that's one big thing. And here, being part of the SAG, you get an oversight of the whole range of schemes that are being organised. So it gives you quite a broad knowledge. So it's a learning experience really, because you're continually learning about other areas that you might not have so much expertise in, and then the areas that you do have expertise in, you're seeing the range of reporting, or the range of reports that are out there for that area. So yeah, it's good. and it's a great group of people as well to work with, so that's really nice. So I should say that we are often looking for new assessors, either as to join the SAG or as second markers, and if anyone's interested in that, then they can contact the GenQA email address. That looks like it's info@genqa.org. Hope I've got that right. So they're always happy to take queries.
Great, thank you, Carl. And I would just like to add that personally, I've been doing this for many years now, and each year we still learn new things and see different things. So it's fun, I must admit. So we've got quite a few other questions coming in here. One of them is there a plan to join molecular and cyto? So I think that's probably continuing on from the merger of the two EQA providers, the molecular genetics scheme and the cytogenomics scheme, to create genomics quality assessments, so GenQA, and we're slowly working through our different EQAs, the disease-based EQAs, to make it more patient pathway focused. It is something that we have been doing with our molecular pathology and hematology EQAs, and it's something that we're now looking at for molecular and cytogenomics. So for example, we have two currently pilots running that are looking at this type of assessment. One of them is the severe intellectual disability pilot, and the other one is the disorders of sexual development, whereby we're not being prescriptive as to what types of tests or reports are written. It's more about the following the patient pathway, and it doesn't matter whether a test is performed in a traditional cytogenomics lab or a traditional molecular lab. It's about the clinical service. So hopefully that's answered that question. That's the way of travel at the moment. We also have one here about how many labs participate in each scheme and how international is GenQA. So maybe Becky, could you cover that one? I know we've been looking at maps and heat maps of the globe to see where we have coverage. Do you want to touch on that one?
Yes, thank you, Sandy. So we are very international, I think, is the short answer. We have, for the molecular genetics ones, we had 165 labs participated in 2019. Of those, 38 were within the UK. So the rest are all spread across the globe. We have participants in over 80 countries. So hopefully that answers your question. We are very international.
Thank you, Becky. And just to add to that, actually looking at the attendee list here, we have quite a few colleagues from out with the UK. So welcome. I think that just echoes what you've just said. I think looking at the time, we should close very soon. We've got one final question here. So this is, what happens to labs that make errors in more than one EQA in the same year? So I guess this is based on the fact that we have multiple different disease EQAs that running under the umbrella of molecular genetics. So I guess it depends on performance in each disease EQA. Becky, do you want to just cover that issue, please?
Thank you, Sandy. So about performance, so if a laboratory makes an error in more than one EQA in the same year, the first place probably for participants to look at is our performance criteria, which is available on our website. So if they make a similar issue within the same year or even consecutive years, so across different EQAs, the lab's performance will be reviewed by the SAG, and they will generally be classed as a persistent poor performer. And we will provide help and advice to that laboratory to try and assist them in looking at that issue and working out the best way of going forwards to ensure that they can resolve that problem. If they have two different issues, then that would not trigger persistent poor performance. And I think the final thing to say is if the lab is based in the UK or Switzerland and they are a persistent poor performer, then they would be reported to their national performance oversight group. So that's just a quick run through of what happens if they make more than one error.
Great, thank you, Becky, and all the documentation's available for anyone that wants to look at the performance criteria on our website. Yeah, great, thank you. Next slide, please, Jenny. Okay, Farah, back to you, thank you.
Thanks, Andy. So I hope this our first Focus On webinar was useful and you've enjoyed it. We welcome your feedback. As you've registered, you'll receive a survey straight after this, after we close. And we will then, for a limited time, this will be available just to those who have registered, but we will later make it available on the GenQA YouTube channel. So please do subscribe when you visit our channel. And that's how to keep up to date on, we keep, as well as our website where we have the news feed, and we do keep this up to date as well. Everyone who's registered, you'll all receive participation certificates. And just a reminder, next Focus On topic will be clinical genetics. So please do join us for that as well.
Thank you, Farah. So it just remains for me to thank our panel members very much indeed for participating today, and also for everyone who has attended, and we hope you join the GenQA webinars in the future. Thank you, everyone.